Tonight's webinar called the Australian DNA Test for the Pole Gene. This is our 6th webinar session in a course of 6 sessions titled The Cutting Edge, the latest research from the beef CRC. My name's Christian Duff and I'll be your facilitator tonight. I work for SBTS and based in Armidale. The main presenter tonight is Dr John Henshall. He's from CSIRO Animal Food and Health Sciences, and he'll be presenting the main part of this session throughout tonight. And we thank John for his time to put into this presentation. It's definitely going to be worthwhile because it's a very interesting topic that's very important in the front of everyone's minds, particularly in breeds that have horned cattle. I won't steal John's thunder though and talk about what he's going to talk about. I'll now move on and just give you a bit of background behind this series. The Cutting Edge webinar course, as I said, has been running over 6 weeks. This is the last session in this webinar course, as highlighted on the screen there, the Australian DNA Test for the Pole Gene. We've run 5 sessions prior to this, 2 on genetics for reproduction, the male influence and the female influence. They were done by David Johnston from the Animal Genetics and Breeding Unit. The third and the fourth were done by Wayne Pitchford, maternal productivity in the south, relating to those things. And the 5th session was done by Mike Goddard last Monday in regards to the CRC test for growth, feed efficiency, carcass and reproductive performance. And we've got tonight webinars with John. Now, for any of those who missed the last sessions, you can actually catch those up on our SBTS or TBTS webinars page. We've got a link to each of those sessions. They've been recorded, so you can watch them at any time. They basically take you to a clip on YouTube, and you can basically watch it. You can't obviously interact with the producer because it's with the presenter because it's a recorded session, but at least you get the benefit of getting the up-to-date research in front of you if you happen to miss those sessions. Obviously important we run this webinar series because, as most of you would know, the CRC closed its door on the 30th of June this year, but that doesn't mean the really important science behind the research and the research outcomes stop. A lot of scientists and scientists such as John and CSIRO are continually working to make these technologies practical and useful. So it's important we keep up on top of them and keep progressing forward as an industry.
Are you there, Christian? I've clicked. Can you see what you're supposed to see?
Certainly can see your screen, John, so you're good to go.
Okay, thank you very much, and welcome everyone who's here listening and watching. It's I think it's more than a year since I've delivered an update on what we're doing with the Australian test for pole. But so tonight I'm going to go over some of the stuff that we've covered before, but also to talk about some of the new work that we've been doing in this area. Right, beginning again. But I'll begin with the take-home message, and that is that a polled bull may carry 1 copy of polled and 1 copy of horns, and you'll hear to that sometimes referred to as single copy polled, heterozygous, or a carrier of horns, or alternatively that polled bull may carry 2 copies of polled, and sometimes people will say double copy polled, homozygous polled, true polled, or they might say he's not a carrier of horns. And single copy polled bulls will pass on a horned allele to a half of their calves, and even if those calves themselves are polled, they'll still be carriers of that horned allele. And what that means is that if there's a chance that either parent of a bull carried horns, then the only way you'll be able to tell whether or not he's a single copy polled or a double copy polled is either through a progeny test where you join him to cows and look at the results in the calves, or using a DNA marker test. The other half of the take-home message is that Scurs appear mostly in animals that have 1 copy of polled and 1 copy of horned, so in heterozygous animals. In animals that are heterozygous, the frequency of the three phenotypes, that is what the animal actually has itself, so either polled, squared or horns, varies between the breeds. And this variation is genetic, but we don't know whether it's due to a small number of genes or to many genes. Now, if it turns out that it is due to a small number of genes, then we should be able to develop DNA marker tests for Scurs as well. So that's the end of the take-home message, and now I'll move into the background. And those of you who've basically been following poll for a few years will find some of this background you'll already know. For others, it may be new. So just firstly, this week we're talking about a single gene effect. Just single gene effects, basically, where only one gene or a small number of genes causes all or most of the variation that we see. And some really basic genetics, at each location on the genome, each animal inherits one allele from its sire and one allele from its dam. And sometimes the effect of those alleles is additive. That is, that animals with two copies of the same allele get twice the effect of animals with one copy of that allele. Sometimes it's not additive, and that one example of that is dominant. And when the effect is dominant, one copy is enough to get all of the effect. And this is what happens with polled, at least in British breeds and European breeds. Polled is dominant to horned. One copy of the P allele, the polled allele, is enough to make you polled. So if you're homozygous PP, you're polled. If you're heterozygous PH, you're also polled. And it's only if you've got two copies of the horned allele are you phenotypically horned. But that's in Bos Taurus breeds and British breeds, European breeds. It's not the same in cattle of Samuel or Zebu origin. There, if you're homozygous polled, so PP, most of those animals are polled, but not all of them. If you're heterozygous, so you a PH, you have a P allele and an H allele, then you might be polled, you might be scurred, or you might have horns. And if you have two copies of the horned allele, then you're, well, as far as we know, definitely horned. And if you look at this, you can see why it is necessary, the things on the right-hand side there. If we look at the polled animals, so we've got, in Bos Taurus, we've got these animals here that are PP, these animals here that are PH. If an animal is just polled, that's not enough to tell you whether it is PP or PH. And it's the same in the Bos Indicus breeds. You can be polled, even that's why we need a marker test to distinguish between the animals that are homozygous polled and the animals that are heterozygous, the carriers of horns. So the Australian test for polled, the one developed through the BCRC and with funding from MLA, was an alternative to tests that were developed in the Northern Hemisphere in Bos Taurus breeds. The other tests were on the market, but they certainly weren't validated for Australian populations and definitely not validated for our tropical breeds in the north. And so right from the beginning, we were looking for a test that would work across the range of cattle, beef cattle that are found in Australia. And we developed the test in Brahman, but it also proved useful in a range of other breeds. Hereford, Droughtmaster, Charolais, Simmental, Standards and tropical composites. But it wasn't so good in Limousin and Brangus. And a lot of what I talk about, or some of what I talk about tonight, will be the reasons for that and what we've been doing about it. So this question, why doesn't the test, our test? So our test uses a marker, and this again is something that I've talked about before in webinars. Our test uses a marker which is linked to the causal mutation. This means that what we'd really like is a test where we could actually measure the gene that's responsible for polled and horns. But we don't have that. We don't have a test which is actually measuring the causal mutation. We have a marker that is nearby on the genome. So it's linked, but it's not the actual cause. And most of the alleles at our marker are either always associated with the polled genotype or always associated with the horned genotype. But a small number of alleles have associations with both polled and horns. And for these markers, the allele frequencies and the associations with polled and horned differ between the breeds. And we call these alleles ambiguous alleles, amongst other names. And it's the reason that our test doesn't work as well in Limousin and Brangus is because these ambiguous alleles are quite common in those breeds, whereas in other breeds, such as Brahman and Hereford, they're not very common, and therefore mostly our test gives a fairly clear result. So in all of this, there are two sources of variation. Firstly, the underlying genotype is not perfectly linked to the phenotype. That's especially in Bos indicus breeds. So this is, if you're heterozygous, you might be polled, or you might be scurred, or you might be horned. It's not a perfect association. And that also affects the accuracy of progeny tests. And the second source of variation is that our marker is not perfectly linked to the underlying genotype. But we do know which marker alleles are the problems. And I'll talk a little bit more about this later on. So I'm going to pause for questions here, but specifically questions on this inheritance, how the inheritance and the fact that you have the two alleles and the effect of the two alleles. If the questions are about the test or how you might use the test, it might be better to wait until a little bit later on.
Thanks, John. We currently don't have any questions submitted in regards to that particular part of the topic. What we might do is remind people that if you want to ask a question, you can basically type a question into your question pane on your control panel and submit that, and we will throw that question to John at the appropriate time when we answer questions. One thing I'll also add, we're having a little bit of not major problem with John's audio, but occasionally it just slows down a little bit, so just bear with us tonight. I'm hearing him quite clear from my end, and I hope you are as well, but just occasionally it slows down due to the bandwidth probably. We do have a question here for you though, John. Is there only one gene influencing the polleness of cattle, or is there, looks like there's multiple genes? Can you answer that one?
I'll answer half of it, but I'll leave half of it for later in the presentation. Certainly there is one gene which people refer to as the poll gene, which is responsible for almost all of the variation in the Bos Taurus breeds. So in dairy cattle, in Herefords, in British breeds, in European cattle, one gene appears to be responsible for nearly all of the variation. So when people talk about the poll gene, that's the one they mean, and that's the one that our test measures. A bit later on I'll talk more about some of the other possible models that certainly might apply in Bos indicus breeds.
Sure, thanks, John. Just a question here that's quite specific to a breed, but I'll ask anyway, and it probably relates to more. You mentioned the ambiguous allele. Are you going to go through later on which breeds have this ambiguous allele affecting particularly the early stages of the test? This question particularly came from a Devon breeder. But are you going to go into more detail there about which breed that affects more than others?
I wasn't going to, and to be honest, I don't know the answer for Devon cattle. If the breeder has, or if you have submitted samples to either of the genotyping laboratories that use our test, then you might have a better idea of the answer to that question than I do. And I must admit, I'm not familiar with what the actual results are in Devon cattle.
Sure. All right, John. Well, I think they're the questions that are relevant for this part of the your session. So I think we can move on to the next section.
Right. Okay then. So I'm going to talk something more about these ambiguous alleles and what we've been doing about, or what we think might be the cause, and what we're doing to try and resolve this the problem of the ambiguous alleles. And to do this, I'm going to talk a little bit about, well, firstly, our marker test. We have at our marker there are lots of alleles, potentially dozens of alleles. One of these, and these alleles each have a number. You can think of that as the label of the allele. And one of the alleles is the 303 allele. And it's quite a common allele, very common in polled animals, and it's almost always associated with polled. So an animal might have one copy of the 303 allele, in which case our marker test would say it's probably a carrier, it's heterozygous, or it might have two copies of a 303 allele, in which case our test would say that animal is probably homozygous polled. And we can say that because most of the other alleles are almost always associated with horns. So there's a range of alleles, 303 is one of them. It is the one that is overwhelmingly associated with polled. All of the other alleles are associated with horned. But there's another allele, the 305 allele, that is sometimes associated with polled and sometimes associated with horns. And whether or not it is associated with polled or horns seems to vary depending on the breed. In some alleles, in some breeds where 305 occurs from time to time, it seems to be still associated with horns. In other breeds where 305 appears, it seems to be associated with polled. And in some breeds, and Limousin is a good example, 305 seems to go half of the time with polled and half of the time with horns. Now, one of the things we tested was some Angus animals, which we're assuming are all polled. And it turns out that 305 is very common in Angus. And this led us to the hypothesis that maybe if the 305 came from an Angus ancestor, such as during grading up of a breed, then that 305 is polled. But if it came from the ancestral breed, then perhaps it's a horned allele. So to use a Limousin example, Angus were quite widely used in grading up Limousin in Australia. Maybe the 305s, if they came from an Angus ancestor, it's a polled one. But if it came from a French Limousin, perhaps it's always a horned one. And this is a hypothesis that we came up with maybe a bit over a year ago to explain to ourselves what was going on, why are we getting these ambiguous alleles. So once we had the hypothesis, then the next step is obviously to see what we can do about using that hypothesis to see if we can improve the test. One way you could do this would be to trace the 305 alleles through the pedigree and declare them as polled or horned, depending on their likely origin. You could look at the pedigree records for an animal, and if you were to genotype the ancestors of the animal with our test back to the founding population, and especially if you could genotype right back to, say, some cows that were used in grading up, then you would be able to say, well, you can see where this 305 came from, and therefore we could make a call on whether or not it's a polled 305 or a horned 305 allele. A similar thing would be to actually use the results from progeny tested animals. If we had a progeny tested bull that we knew from progeny test results that he was homozygous polled, and it turned out that he had a 305 allele, then we would know that that particular 305, the one carried by that bull, and maybe by his brothers and maybe by his sons, was a 305 polled allele. So that's actually would be quite a powerful way of moving forward. The problem with it is, of course, that it needs pedigree information, and we would prefer that our test worked in all animals, including ones that didn't have pedigree. Well, it's certainly not. We wouldn't want it to be a requirement of the test that you have to have pedigree information as well. So the other solution that we've been looking at is to add markers that are close by. So we have our test marker, our test microsatellite marker. Bulk up the test with some other markers that are close by, and maybe these markers would indicate the origin of the 305 allele. So essentially this means instead of depending on a single marker in a test, we would use more than one marker, and if there were some markers that were that indicated that a sample came from an Angus animal, then we could say, well, in that case, the chances are that that 305 allele at our test marker is associated with polled. And so that's one of the things that we've tested. We tested an additional, well, first about 20 markers, but we very quickly cut that down to about 10 additional markers on about 300 animals from a range of breeds. And all of these animals had the 305 allele. So that means they were ambiguous. We weren't sure whether they carried a 305 to do with polled or horned. But we did know that the animals themselves were either polled or horned. It proved not as easy as we thought it would be to detect alleles of Angus origin. In fact, that really didn't work very well at all. However, the test with about 10 additional markers certainly did show promise. It certainly worked a lot better than just our single marker test that we have. So in this small sample of 300 animals, and these were carefully selected animals because they all had a 305 allele, it turned out that we got very promising results by adding additional markers to the test. We certainly need to validate that on a larger population, and we need to do that across breeds, and we're keen to do that, and we would certainly like to proceed with that as soon as we can. At the same time, other international groups are refining their tests as well, and there's a lot of international effort going into finding the gene that's actually responsible for polled, and we're also engaged in that as well. And so a lot of people around the world are working in this space. And my conclusion from that is that it's very likely that the test will improve over the next few years. We would certainly like to improve our test sooner than over the next few years, much sooner ideally. And we're certainly working as hard as we can to do that. Other groups around the world are working as well. And I think it's most unlikely that we won't have tests that are a lot better fairly soon in the next 1 or 2 years. But that's not a reason to wait, because on most animals out there, the tests that we have at the moment, it works quite well, and it's a lot better than no test at all. And you can put this into perspective by looking at the test performs the same functions as a progeny test for polled, but you get the answer a lot earlier in the animal's life. And I'd certainly encourage people to use existing tests, either our test or other tests that are out there, if you're selecting polled, rather than wait for the improvement that I'm saying is likely to happen, but I can't guarantee it. So when we look at these progeny testing, and this is just a reminder about our tests, not necessarily the other tests, if you have a phenotypically polled bull and you're joining him to horned cows, the marker test result of homozygous polled PP is about the same accuracy as seeing 5 out of 5 calves polled, and that's that's reasonable. It's not It's not a guarantee that he's a pole bull, but it's a pretty good indication that he's homozygous polled. In European cattle, if you saw one horned calf out of 5, that would be fairly good evidence that he was heterozygous. But that wouldn't be the case if the cattle had some Bos indicus content. One horned calf out of 5 in that circumstance is fairly weak evidence that he's heterozygous, because we do see examples of where homozygous polled in Bos indicus cattle sometimes might produce horns or scurs, and you would need a much larger progeny test to get the same amount of accuracy in those herds. And that really leads us on to this question of scurs and also the African horn gene, and there was a question from a listener earlier tonight. Are there other genes out there that affect polled? And I've said several times tonight that in cattle of Bos indicus cattle we see mostly polled if they're homozygous PP. If they're heterozygous, then we see polled, we see scurs, and we see horned. And homozygous horned, we always see a horned phenotype. And we know that this variation within those heterozygous PH animals is at least in part due to genetics. But we don't really know whether it's due to one gene or to many genes. And if it's due to one or two genes, a small number of genes, are they additive, dominant, or recessive? What's the mechanism by which these genes work? And the African horn gene, which is a hypothesis, really, it's not a gene. We don't know where that gene is on the genome, but it's a plausible explanation for some of the variation that we see. Perhaps that information is out there in industry, in fact almost certainly, because a lot of people are joining polled bulls to horned cows in herds that have Bos indicus content. And all you'd need to do really to answer these questions is to measure the phenotype on all of those calves and what proportions are horned, what proportions are polled, what proportions are scurred, and then follow that for 1 or 2 generations through the pedigree, and you would know the answer to this question. The problem is, of course, that we never get that phenotypic information because the calves are dehorned before the phenotype is clear, and that's the standard procedure, and it's quite difficult for us to get accurate information on what would the phenotype be on adult calves from herds where people are joining polled bulls to horned cows. So consequently, we're looking for ways of capturing that information from other sources, and there are a lot of genomics experiments going on in Australia, and the beef CRC has been a major driver there. You will have heard about some of these last week and in previous weeks. A lot of animals have been genotyped for slip chips, and we're certainly keen to try and use this information to get a much better idea of the answer to the questions that we're talking about there. And it is important because if it does prove that 1 or 2 genes is responsible for scurs or for horned animals in animals that are heterozygous of the polled gene, then we would have to be able to have tests that would allow us to say with a higher degree of certainty what the result of using this particular bull would be over these particular cows. And at the moment, once we get into the Bos indicus breeds, we really can't do that. All we can say is things like there will be a high proportion of scurred progeny. Whereas if we had tests for these things, then we could give you an answer and say this is what you'll get if you join this particular bull to cows from this background. So we're certainly alert to the possibilities of using data from existing experiments to help us out there. So I'm going to break for questions a second time, and this time certainly more about these ambiguous alleles, improving the test, scurs and the African horn gene. But I'll also let you know that what I'm yet to talk about is how you use the test results in a breeding program for polled. So if there are questions that relate to that, maybe it'd be better to wait.
Thanks, John. Actually, most of the questions we've got a few here relate to how you use the test. For example, what does 90% PP actually mean? So are you going to be covering that in the next part?
I think it's in about 1 or 2 slides from here, and I'll deliberately focus more on it, knowing that those questions are there.
Yeah, okay. I think we can probably. There's no late questions coming in, so most of the questions I've got relate more to how you use the test and breeding for polled. So I think it's probably best to go on to your next part of your presentation, and we can throw those questions up then.
Okay, I'll move on. So breeding for polls. DNA marker test for polled is currently offered by 2 commercial providers, and the format of the results might differ, but the results are based, because the tests are essentially the same test, so it's using the same information, so really you're getting the same things from both providers, but perhaps in a different format. And when we do report results, it's actually quite difficult to decide on the best way to do it, because you have to balance 2 conflicting needs. First, you want to keep the message simple, and we don't want it to be so complicated that people can't use the test. We want a person to be able to look at a test result and to have a feel for what that means, so that they can go out and use that test in their breeding program. But at the same time, we don't want to pretend that the test is perfect, because we know that there is uncertainty in the results, and we certainly want to be honest about that, and so we want the tests to reflect the degree of certainty that we have in the results that we're providing. And 2 of the options that you might see are, one option is to report probabilities of genotype classes. So at the polled locus, the polled gene, animals can only be homozygous PP, 2 copies, true polled, heterozygous, so they're carriers, or homozygous horns. That's the only 3 possibilities. And there is a true underlying genotype that animal is, but we don't know what that is. We have a test result that tells us with some degree of certainty what that underlying genotype is. And in this first case here, so if we look at this example here, we're saying that there's a 5% chance that he's homozygous PP, a 94% chance that he's heterozygous PH, and a 1% chance that he's homozygous horned. Now, reading backwards, what this tells you is that the alleles that he carries are the alleles. One of the alleles is one that is almost always associated with polled, and that would be almost certainly the 303 allele that I mentioned before. And the other allele he carries is one that is mostly associated with horns, but perhaps we have seen 1 or 2 examples when it was associated with polled, and that's why the probability that he's homozygous polled is not 0. It's a number that reflects the fact that his second allele is one that very occasionally might occur in animals that are homozygous polled. However, we're pretty sure that he's not homozygous horned, and so the 1% there reflects the fact that we know that animals with a 303, that 303 is almost always associated with polled, and therefore it's very the second way of reporting the results would be, in this a different example to number 1, is to just give a probability that he's just give his most likely genotype class and some measure of the accuracy. And it might be something like heterozygous. The most likely result is heterozygous, and we'll call that with 92% accuracy in this particular case. And again, that will be based on the marker alleles that animal carries, possibly also the fact his own phenotype. If it's reported that he's got the marker alleles he carries might give us 90% or 92% case in this certainty that he's a heterozygous animal. Now usually those two ways of reporting results are equivalent. The possible exception is when an animal has an ambiguous allele. So in that case you might see a result where, if we look at this top example here, instead of being 94% in here, it might be something like 44%, and with also a big probability of it being homozygous polled. And what that's saying is that he has probably again the 303 allele, but also this 305 allele that really doesn't tell us much about his second marker. So in breeding, well, I wasn't going to pause then, but maybe I should while I've got that slide there, given the questions that that we have.
Sure, John. The question relating to that, maybe you've answered it, but just to clarify, the question was, what does 92% PP mean? Does this mean 92 out of 100 progeny will be polled? And I guess I can short answer that and say it does mean that, but you just want to clarify what that 92 means again, 92%?
It doesn't, no, that's not what it means. It would mean if you had, because the bull has a single underlying genotype. He is either homozygous polled or he's heterozygous. The problem is that our test can't tell with certainty which of those he is. And so it's not the case that 92% of the time he'd pass on, let's say, polled alleles to his progeny. It'll either be 100% of the time or 50% of the time. It's if in a theoretical way or a hypothetical way, if you were to buy 100 bulls and each of them were 92%, then in 92 out of those 100 bulls, you would get what you think you were getting, that might be a homozygous polled bull, but in 8 out of those bulls, you get the other alternative, the heterozygous bull. So it's our best measure of the uncertainty associated with the prediction. But in reality, the bull is either one or the other, or one of those three genotype classes.
Thanks, John. The other question in relation to this, and maybe it will be covered later, but basically, as you know, there's some labs in the overseas like GeneSeek and Igenity who have a multi-marker test on the market, including horn polled, whereas they seem to be able to provide a higher level of animals with a 90% heterozygote PP, homozygote PP, for example, and less of these more ambiguous results like 47.5% probabilities, which are pretty hard for breeders to work with. I guess what's your thoughts on where those particular breeders should go to I guess get the DNA information that they're after, or the results that they're after, which are higher accuracy results?
Well, the answer is that certainly if you are of a breed where ambiguous alleles are common with our test, then, and if there is a test on the market that has been validated for your breed and that does provide a higher level of certainty, then certainly you should. I'd encourage you to use those other tests if you're breeding for polled. We're certainly aware of the deficiency of our test in that area, which is why we are working to try and improve it, and to improve it so that it works across all breeds in Australia equally well. I mean, our internal target is 99%. That's what we'd like to get. But until we do get to there, if there is a test out there that does a better job than our test, then certainly, and assuming that it's available and it's validated in your breed, then most definitely you should be using it.
Thanks, John. I think they're the related questions there, so feel free to move on.
Okay. Talking now about this process of moving your herd to polled. So I'm assuming here that you're using the test. Homozygous polled bulls are preferable to heterozygous polled because, as we've described, they're going to pass on twice as many polled alleles as the heterozygous bulls will. But that's not enough, because single trait selection is almost always bad. You already know what's important to you, and you need to consider all of the important traits in your breeding objective, not just polled. And the best way to do this, of course, is to use EBVs that I'm assuming that you're already familiar with, and that allows you to rank bulls. Then you have this second thing, which is the bull polled or not, and you then have to balance how much value do I put on the polledness compared to these other traits. But you certainly can't afford to ignore the other traits that are important to you. And the optimum strategy to bring polled into your herd really depends on the availability of good polled bulls for your breed. Now, if there are lots of these available, if polled is common in your breed and there are plenty of bulls available and they have the genetics that you want, then you should use them. And in that case, it's not a difficult question. There's an easy answer. In the breeds where polled is much less common, where it's difficult to source polled bulls, and maybe it's difficult to even get homozygous polled bulls because there aren't many out there for sale, and the ones that are there are expensive, then it's a lot more difficult. And if you're a bull breeder yourself and you're trying to get polled into your herd, you really need to think of it, and there's a word for this, and it's called introgression. You want to introgress polled into your herd while losing as little as possible of the other good genetics that you already have. And this makes it a more difficult task, because you might be prepared to tolerate heterozygous bulls for quite a few generations while you fix the other good genetics that you have in your herd into that line of polled bulls. And in doing that, that means that you might have to use a marker test for several more generations than you would if you were prepared to just do essentially a genetics replacement with polled bulls. And so it's a much more complicated strategy, it's going to take longer, and you're going to have to keep using the test for longer. But if you do that, and you do it in a careful way, you can reduce the impact on the other traits that are important to you. The other aspect to this is dehorning, because remember, the reason we want to breed for polled is to reduce the need for dehorning. And in commercial herds, you can consider strategies that will reduce the need for dehorning in the short term. In Bos Taurus herds, because very few progeny of a homozygous polled bull will require dehorning regardless of the cow type, this isn't very hard. If you get homozygous polled bulls, you've already essentially eliminated the need for dehorning in your commercial calves. But it's less clear-cut in cattle of Sangor or Zebra origin, because of the fact that in these heterozygous animals we get animals which are polled, we get animals that are scurred, we get animals that are horned, and at a young age you may not be able to tell the difference between scurs and horns. So even if you were prepared to tolerate scurs and not dehorn, you still don't really know for sure what you're going to get. You might have to, it might be quite difficult to quickly reduce the need for dehorning. And this is why we would like to have a much better idea of the other genes that are affecting it, so that we were able to give some sort of degree of certainty about what will the progeny of this bull over these cows be. If we know that they're overwhelmingly going to be scurred with no horns, then it might be that some people would choose not to dehorn in that circumstance. Other people would still dehorn in that circumstance. It might be that you consider a terminal cross using something that you're pretty sure will not produce any horns over the cows, but not keep the heifers, because you know that those heifers aren't really what you want moving forward in the long term. And really there are big gaps in our knowledge here, and some of the people who are out there using polled bulls over horned cows will have possibly a better idea than we do about what actually happens in the various circumstances when you use different sorts of polled genetics over different sorts of horned cows. So I'm going to come back to the take-home message before I finish. And I think that we've been over this many times tonight. Polled bull may carry one copy of polled and one of horns, or he may have 2 copies of polled. The single copy polled bulls will pass on horned alleles to half of their calves, and those calves themselves will be carriers. So if there's a chance that either parent of the bull carried horns, then really if you want to know whether he's homozygous polled or heterozygous, then you don't really have many choices but to do a DNA test. But the light at the end of the tunnel there is that, of course, as you use more and more polled genetics, the chance that either parent carried horns is reduced, and so eventually you can stop testing. That's it. Questions?
Thank you, John. Got quite a range of questions come in, stretching across the whole spectrum of what you could think about in regards to horn and poll, so bear with me while I put them together. Just in regards to I suppose individual breeds that now are saying, well, what sort of level of accuracy will I get out of my breed for this test? Does this ambiguous thing affect me? For example, as a blonde Aquitaine breeder asking the question, I'm sure some other people are thinking that, as in, I'm at a stage where I'm comfortable investing in the test in the Australian labs as developed from the CRC. Where are they best to get that information? They're probably better off just talking directly to the labs that offer the test, are they?
Certainly, if the labs have offered the test and have genotyped animals in that particular breed, then the labs will know the answer to that. They may not be providing that information back to us. I mean, even though we work with them, it doesn't mean that we have access to all of their data only, and so it's likely that I'm not going to know the answer to that particular question for any particular breed. If the labs have never tested any animals of that breed before, then really the only way to find out is to send in some samples and do the test. And if it turns out that you are finding a high proportion of ambiguous alleles, then your options are either go to a different test from an overseas provider, or wait until we improve our test, which is certainly we're very keen to do that, and we're optimistic that we will be able to do that in the reasonably short term, but of course we can't guarantee that.
A question here from someone who's obviously interested in this area, basically saying, is it correct that overseas researchers have discovered the actual causative poll gene? Have you heard that at all, John?
There have been some recent papers in this area where people have sequenced the genome of animals, polled versus not polled, and certainly people are finding very strong associations. No one that I'm aware of is yet claiming to have found polled gene. They're finding things that are very predictive of polled or not polled across a range of breeds, but none of them are going as far as to say this is actually the cause of the causal mutation, the thing that is responsible. This is the mechanism, this is how it works. There are perhaps reasons for that. The burden of proof to say that this is actually the thing that causes the effect is quite high, and in case of horns, you can't go and do a test in a model animal such as a mouse and prove that if you make this change the animal does or doesn't have horns, because mice don't have horns. So it's actually very difficult to prove, and certainly, and this is why I said that I expect that there will be highly accurate, more accurate tests on the market fairly soon, regardless of whether, in our case, we're successful with making our test better, but because on the basis of these results that are coming up from other labs.
Thanks, John. Just in regards to the whole scurrying effect, there's a few questions in relation to this. Obviously you can have a herd of cattle which are quite polled in regards to the horn poll status based phenotypically and genetically, but you can still have this scurrying issue come through. Is that something that is being looked at in more detail along with this research, not just at the horn poll and the ambiguous allele effect, but also can we get a better genetic description of the scurrying outcome?
Most definitely. The work that we're proposing to do now has two components to it. First of all, making our test much better and with fewer or preferably no ambiguous alleles whatsoever. And as I said, the target we have is 99%. But also at the same time, we recognise how the existence of Scurs really does make it difficult for breeders in the north, especially, to come up with a strategy that works for them in the short term. And so most certainly we would like to be able to tell you that these are the reasons these animals have Scurs, and once we understand that, then we can test for that as well. So the answer is yes, it is definitely of interest to us, and it's something that we certainly plan to do.
Yep. Thanks, John. Probably a pretty simple question here, but one that's very important is how do you actually get the DNA testing done? What's the process? Basically, and John can add to this, but I'll add my two bobs worth. There's 2 labs in Australia at this stage that offer this DNA test, and that would be the University of Queensland Animal Genetics Lab based at Gatton in Queensland, or Pfizer Animal Genetics. So you can basically go to those 2 Australian labs and get this particular licensed test done. Similar to other DNA tests, you need to get a sample kit from those particular labs. Obviously, they prefer tail hair, so pull tail hair with the roots on, and from that they extract the DNA and utilise in this particular test. Do you want to add anything to that, John? Fairly simple but important.
No, I don't think so. Obviously, if you already have a relationship with one of those labs for other things, for parentage, then you might go with the one you're already used to. But no, I don't have anything to add.
Okay. Well, there's some other questions there, and I apologise for time we might have to pull up, but I encourage you to get in touch with your allocated technical officer for your breed, which I'll put up on the screen in a second. But what I'll do now is I'll just hand the presenting role back to myself, and please don't go away. I've just got a couple of jobs for you to do before I finish up, so I'll put the presenting role back to myself now, John, and I will continue on with a short poll. So everyone, what I'm going to do now is actually going to collect some of your feedback from tonight's webinar, and this is pretty important to do. It's good for our feedback to a range of our stakeholders. So the first poll I'm going to put up in front of you is quite simple. I'll just launch that now, and it will come up in front of you in a couple of seconds. That webinar, that poll, as you can see, is did you find this webinar useful? So you can just take a second to answer that. You can strongly agree, unsure, disagree, or strongly disagree. So we've currently got about 70% of people voted. Wait until we got about 80 or 90%. That's come in, and I'll close off that particular poll. And I've just got a second poll for you. And I'll launch that now. And basically that second poll is based on this webinar. Do you know more about beef CRC outcomes and how they can be utilised in your enterprise? So again, strongly agree, unsure, not applicable, disagree, or strongly disagree. And we thank you for your putting an effort in voting. It's pretty important for us. Right, we're up around 83% voted. I'll close that off in a second. 86%, great. I'll just close that off. So thanks for your time there. We'll now go back to my screen. At this webinar, we can it'll be made available for viewing from the SBTS and TBS websites and from YouTube. If you just go to websites, it links to YouTube anyway, and all the previous webinar sessions are there as well, so you can view those at your own leisure. If you would also prefer to, and if your bandwidth's an issue, then you can also request a CD containing all the webinar course and the presentations, and we can get that to you, so just send us an email. And as I said, there's quite a lot of unanswered questions tonight, so I encourage you to get in touch with your allocated technical officer through SBTS and TBTS. They're up in front of you there now, being Paul Williams for the Tropicals, Andrew Byrne for Hereford, myself being Shorthorn, Charolais, Red Angus, Simmental, Wagyu, Gemma being the other breeds listed, Peter being Angus, and Alex being Limousin. So it would be very remiss of me if I didn't also, before we concluded tonight, thank John for his time. It's a lot of effort to go into to present these results, and they're definitely worthwhile. So from everyone, on behalf of everyone that logged in tonight, John, thank you very much. And I'd also like to thank everyone who logged in tonight, but also a lot of you logged in for the full 6 sessions, so well done on that, and I trust you learnt a lot from the sessions. But I encourage you, if you've got any questions, to contact your SBTS technical officer. So thank you, everyone, and SBTS and TBTS will be communicating with you in other means, but this is the end of this webinar course for this point in time. So everyone have a good night, and you'll hear from us. Good night.